Good day. In this lecture, we will be discussing about multiple sequence analysis. So first of what is MSA or multiple sequence analysis? Basically, it's doing alignment, but instead of pairwise, you are doing it multiple times, or rather involving 3 or more sequences. So a multiple sequence alignment is basically a collection of 3 or more proteins or nucleic acid sequences that are partially or completely aligned. When we say partially aligned, because we are actually looking at here as one of the usual mode here is local alignments. Anyway, uh, MSA usually done to look at the domains and motifs in protein families. So domains or motifs that characterize a protein family are defined by the existence of multiple sequence alignment of a group of homologous sequences. And that is why we were able to find or establish protein families. Because when we aligned different proteins, there are patterns, basically quote unquote patterns that arises. Basically, these patterns are the domains. These are regions in the proteins that are aligned very well with one another. Although even if the rest is not as good an alignment as it is, you have domains or regions where there's a really very good alignment. And most of these domains are characterized by specific functions in that particular protein. An example for that, we can look at certain domains that holds the heme group in the globin protein families. So basically, the globin protein family has a domain that contains their that its function is to hold the heme group, because globins are known for having heme groups that where your oxygen binds. Example for the globin is the hemoglobin subunits, the alpha and beta subunits, and also we have the myoglobin. So that's a domain or a motif. Other domains and motifs, especially for the DNA binding proteins, you will see their regions or domains in these proteins, even though they are not really at first glance they don't really look the same, or they don't seem to have much connection, but they are DNA binding. You will see domains there that that are very similar to one another and that are used to actually bind to the nucleic acid for DNA binding proteins. So these are the domains and motifs, and we can see them through the use of MSA. Now, there is not necessarily one correct alignment of a protein family. It's basically just, well, because MSA MSA itself, there is really no correct or highest scoring in the MSA. When you do analysis on MSA, you are, rather than looking at the score, you are looking at how a certain amino acid or certain groups of amino acids are seen to be highly conserved on that particular class or that particular group that you are aligning. And that is MSA. So it's not really strictly in a numerical sense. Now, here is an example of a multiple sequence alignment. This is a portion of glyceraldehyde triphosphate dehydrogenase, or GAPD3H, from 13 different organisms. So you will see here multiple sequences from multiple organisms, basically 13 organisms, 13 sequences from each from different organisms, and they're aligned. Not as perfectly as one another, but multiple alignment. So this is what a characteristic alignment is. This is just a portion of that protein. That doesn't necessarily mean that the full protein is very similar to one another. Not. This is just a portion where it is highly conserved regions. As you can see, there are this arrows here contains some conserved amino acids. And so you have, for example, this isoleucine, and then a little bit of threonine here, but there's a valine, and then there is a leucine here, leucine, isoleucine. So anyway, so these regions, you will actually see here regions where, like this one, MTT. So regions where they are highly conserved, and then there are also some variations in some aspects. This is because, of course, certain mutations and substitutions occurring, or rather, because they are, of course, coming from different organisms. So each organism has a different, its distinct biochemical tendency, or biochemical, how should I say, environment with one, if for their own. Then you have this GAPDH. So GAPDH, just a brief background, GAPDH is an enzyme that is involved in glycolysis. So since glycolysis is ubiquitous across all organisms, almost all organisms have glycolytic pathways. It's actually very almost universal across all species. You would have all, of course, since it's universal, all organisms having glycolysis have this GAPDH. And so you will see here this, because they perform the same function, they have very good alignment or very good conserved regions. Just there are some variations here and there, but all in all, good variations. So in MSA, we are looking at basically not the alignment score, but how well conserved these regions are. So some amino acid characteristics seen in MSA. So aligned columns of amino acid residues may be determined due to features of amino acids, such as they are highly conserved residues, like cysteines that are involved in disulfide bridges. There are also conserved conserved motifs such as transmembrane span or an immunoglobulin domain. So when we say these conserved motifs for that, especially a case in point is in the transmembrane span. You will see here, you would know if a protein is a transmembrane protein. When you say a transmembrane, it is a part of it spans across the cell membrane. And if you remember, the cell membrane has a region where there is hydrophilic region outside and hydrophobic region inside the membrane. It's a double layer membrane. Now, how do we know that there's a transmembrane region in that protein? Is because if you look at the sequence of those proteins, you would see here an alternate. There's a region where there are mostly hydrophobic amino acids, followed by hydrophilic, then another hydrophobic, hydrophilic. So there are alternates. The alternate occurs if basically the number of alternating hydrophobic, hydrophilic, hydrophobic, hydrophilic is dictated by how many times the transmembrane motif spans across, or how many times it zigzags the membrane. So, or if it has an anchor, you would see here a region where there's a consistent hydrophobic region. So those are transmembrane because, of course, it's made to be inserted across the membrane, which is hydrophobic inside. And there are also conserved features of the secondary structure of the protein, such as residues that contribute to alpha helices, beta sheets, or transitional domains. In fact, before, one of the main cues for or rather one of the main foundations for determining or predicting protein structures, three-dimensional structure of proteins based on just the sequences alone, is by looking at conserved domains that have a higher tendency to form alpha helix, beta sheets based on multiple sequence alignment results. However, right now, the most accurate one is actually done by an AI. So an AI basically AlphaFold from what is that? They have developed AlphaFold, which is an AI that can predict now protein structures based on just the sequences. It has been a dream by many structural biologists for decades, and well, we have now this AI. Anyway, that's just a different story altogether. But anyway, going back to MSA. Now, these patterns or regions in MSA, we can also see regions with consistent patterns of insertions or deletions, regions that are highly conserved, and regions that are not highly conserved, or there are regions that are very mutable. You have many variations when you align them. So we will see these regions where there's not much changes, regions where there are lots of changes. Now, here's an example of domains and motifs that characterize a 14 family defined by existence of a multiple sequence alignment of a group of homologous sequences. So here is an example of an alignment. These are conotoxins. These are short, very small. As you can see, this is the whole protein. Just 12 to 13 sorry, this is around 20, actually less than 20 amino acids. These are very short peptides, proteins, polypeptide chains. These are toxins obtained from the cone snail, the venomous cone snail. These are their toxins, and they are known to be very there are many variants of these toxins. So you have a lot of different types of toxins. One of them most diverse group of toxins. However, here is a multiple sequence of alignment of that, and there are some patterns here. Here is one subgroup of those toxins, which contains what we call the type 1 framework. The type 1 cysteine framework is characterized by this disulfide bridges. As you can see, this is their three-dimensional structure. When you align them, you will see they form, they actually align pretty well. They have the same structure. And you will see here that this disulfide bridges, the yellow lines here, this characterized, this is what gives this alpha conotoxin, type 1 alpha conotoxins, their shape, their characteristic shape, because of the existence of these cysteine bonds. Even though if you look at their sequences, you will see variations, especially this one is highly variable. So you will see some patches of some conserved, but all in all, there are really regions where there's really large variability, but you have a consistent occurrence of the cysteines. And that is what makes this protein certain conserved regions. You have these conserved regions, and there you have these hypervariable regions of your framework. So why do we do this? What are the studies we can do with MSA? So if a protein or gene that you are studying is related to a larger group of proteins, so this group membership can provide insight to its likely function, structure, and mutation, basically, evolution and mutation of that protein. In fact, we can actually monitor the strains of COVID, the development, the mutations, and the development of the different strains based on multiple sequence alignment. When we do multiple sequence alignment, especially if the spike protein, if you do MSA of the spike protein of the COVID variants, you can actually see some patterns in which particular part is being they're experiencing a lot of mutation, or a tendency to have the mutation, where is it tendency not to have a mutation. So most protein families have decently related members. Therefore, MSA is a far more sensitive method than pairwise alignment to detect homologs. Because when we do pairwise alignment, we are actually just looking at the scores. Not actually only looking at the scores, but the score of a pairwise alignment is actually a huge it's actually has a large impact on saying if these two proteins or two sequences are comparable. But if we do MSA, when we compare it to a host of many different proteins, we will see a pattern emerging where it is consistent. A certain region is consistently seen across all different proteins, and that is multiple sequence alignment. Also, we can do it when the output of a database search is examined. A multiple sequence analysis format can be extremely useful to reveal conserved residues or motifs in the output. As again, the same with bullet number 2. So you would see conserved regions in multiple proteins. And one of the more important applications of MSA is detecting deleterious variants. So when we say deleterious variants, basically, when a mutation occurs in a certain protein, that mutation can either bring harm, can bring additional or advantage to that organism, or it can do nothing at all. Now, if it's harmful, such as, for example, genetic diseases like sickle cell anemia, it could have deleterious disadvantage or deleterious effect on the organism. It can cause the organism to have malfunctioning protein, hence you would have a certain diseases. Especially metabolic diseases or metabolic syndrome. Those do not exactly, the issue with metabolic diseases, it's a lifestyle change, and most of the time people with, especially the more dangerous ones, they don't really reach adulthood, usually die off early, and there's not really any cure for that, except, well, redoing its genetic information. But in order to do that, you need to start from the very first, the embryo itself. But anyway, and basically with MSA, we can detect now, or we can pinpoint if there are some harmful mutations that occurs in a certain individual that allows him to have diseases based on our multiple sequence analysis. Because when we can see it among other sequences, this deleterious variant, this mutation that occur is change it made it different from the rest of the group. So that means there is what would be its effect of that change. So also analysis of population data can provide insight to biological questions involving its evolution, structure, and function of your proteins of interest. In fact, when doing molecular biology research and even bioinformatics, MSA is MSA is one of the first steps we do in doing this research. And then almost always, not every time, but it's almost always one of the preparatory steps. And also, when the complete genome of any organism is sequenced, a major portion of the analysis consists of defining all protein families to which the gene products belong. Why do we want to define the protein families? Because these sequences, we don't really know. When we do have the genome, we don't really know what does this sequence mean. So if we do MSA, we can now see it which protein family this particular sequence belong to, which give us an insight what this gene is doing, what is this gene for. So database searches effectively perform multiple sequence alignment, allowing comparison of each novel protein or gene to the families of all other known genes. And phylogeny algorithms begin with multiple sequence alignment as the raw data. So phylogeny is the study of basically how species are related to each other. We usually construct trees here. So these are phylogenetic trees. It's much like your family tree. So you have your which species much closely related to another species. So based on the theory of evolution. So, and the foundation for that is doing this multiple sequence alignment. Also, regulatory regions of many genes contain consensus sequences for transcription factor, binding site, and other conserved elements. And these regions can be identified based on conserved non-coding sequences that are detected using MSA. So we can only see conserved sequences, or we can only say that this certain region of the sequence is conserved if we observe it across multiple, or rather we observe it happening across multiple sequences. And we do that using multiple sequence alignment. So we have here 5 main approaches to MSA. So we have first these approaches. So pardon my, let me hide my screen. Okay. So these 5 main approaches, we have the exact methods, and then we have the progressive alignment. Now, the exact method here, why do we have these 5 approaches? The first one is the exact method is the ideal version of the method, but we cannot do that. It's really not feasible to do for reasons that I will explain later. And so we have this progressive alignment. Now, for the progressive alignment, the representative for that is class LW. So it's one of the first one to be developed, but there are also some disadvantages for this alignment. Therefore, we have now improvements on that one, as seen by the development of the other 3. The iterative approaches by Praline, Interalign, and Muscle. And then we have the consistency-based methods by Math and ProbCons. Also, we have the structure-based methods, which includes information about one or more known 3-dimensional protein structures. So among all of these 5 structure-based methods incorporates information from the 3-dimensional structure, but more on that later. So let's look at exact approaches first. So exact approaches is just like the pairwise alignment where we have the matrix, but taken with multiple sequences in mind. So it's basically just applying the dynamic programming that is similar to the Middleman and Wunsch programming. But instead, because remember, in the pairwise alignment, you have the 2 sequences being aligned. You put them in a 2-dimensional matrix. So now the thing with this, if you have a multiple alignment, you would now have 3 or more dimensions to your matrix. You need to add more dimensions in order to have a better alignment. So if you have 3 sequences to align, you would form a cube, basically. So X and the Y axis for each of these screen, and then you have the third in the Z axis. And then you would try to do the diagonal alignment based on the diagonal of the cube. Now that's for the 3-dimensional. What about the 4th dimension when you add it? So basically, if you can visualize how very complicated this becomes. So although these exact methods can generate optimal alignments, they are not technically feasible with our time and space that we have. Because when we want to align, for example, n sequences, the computational time that is required is O times 2 raised to n, L raised to n, where n is the number of sequences and L is the average sequence length. So it is you are actually basically, this is an exponential of the number of the sequences. This is for the computational time. Now, the exact multiple sequence alignment basically has it really not feasible because it would allow us to spend too much time. And there are many important things to do compared to MSA. We want the results. This is just most of the time this is just a preliminary step in our research. We can't spend too much time on this one. So that is why we have developed more methods, such as the non-exact methods, which are the 4 other methods. So here's the first non-exact method developed. This is proposed by Fitch and Yasunobu in 1975. And this is also the most commonly used algorithm. So we call it the progressive alignment because the strategy entails calculating pairwise sequence alignment scores between all the proteins or nucleic acid sequences being aligned. Then we begin the alignment with the 2 closest sequences and progressively adding more sequences to alignment. Now, we call it progressive because we start with, here is your basic steps. So you start with calculating a guide tree. So how do you calculate a guide tree? You do that by first doing pairwise alignment. So at the very foundation of this one is doing pairwise alignment between all sequences. So A to B, B to C, C to D, A to C, A to D, one versus everyone else, basically. And based on the scores, we would form the tree. This guide tree is formed based on which amino acids or which sequences have a closer score with one another. For example, A and B has the highest score, or rather A scores would be the most. So A is first put with B. E has the highest score with F. I has the highest score with J. So they are paired like that. And then with this one, C scores higher with both B and A. And so it is put in here. So basically, this is the guide tree. So G scores the next highest with, or rather, G scores the highest with F and E. So it's put here. Or rather, for example, between sequences E, F, and G, E scores the highest with F, and then the second highest score of E is with G. So it's put like that. So that is basically how you build this tree. Now, based on this tree, you are now going to build up your sequence alignment. So the first step is align first the sequences with the highest pairwise alignment score with one another, A and B. We have E and F and I and J, and then add the next highest scoring sequence. So you have the C, the G, and the I and J, and then continuously add one another until you have aligned all of the sequences. And basically, this is your progressive alignment. It's called progressive because you are progressively adding more sequences. Now, the process for this one is it's a rapid alignment because you're just doing pairwise alignment, although there are many pairwise alignments that you would be doing, but you're just doing pairwise alignment instead of doing multidimensional matrices for the exact approach. But the cons here is the final alignment depends on the order in which the sequences are joined. So if you have, for example, for example, A scores the highest with B and scores the second highest with C. However, what if C scores the second highest or C scores the highest with F, but F scores the highest with E and F scores the second highest with G? Now, based on this tree, since C was added to A and B, so you won't see it even if C has the highest score with F. It would still be clumped together with A and B rather than with F. So basically, that's the issue. Therefore, if it is not an optimal, the order is not optimal, you would have basically this not exactly a mistake, but a suboptimal result that would occur. So basically, it's just if you made a mistake once, it would persist. It might even compound with the next progressive step. So that's the cons. That's the problem with your progressive alignment. But still, for MSA, it's basically a good enough alignment. You can actually have a reliable result here. So this, and we can see that because it's still being used today by Class LW, MAF, ProbCons, MASL, and TCOFE. As actually you can see here, there are some overlaps among these algorithms because you can basically choose what type of algorithm you want to use. Okay, so here is Class LW alignment. This is their web page. You can do it in the web. So this is the steps on how you do that. So global alignment, you have pairwise alignment first, you do a guide tree, and then you progressively add the sequences, align the sequences progressively based on your guide tree. Okay, now when we talk about the guide tree, it's inevitable that we will talk about distances. So the Feng and Doolittle progressive alignment algorithm, it converts similarity scores to distance scores, which is used to generate the guide tree. And this is the conversion. Now, the distance versus the similarity scores. This is from the pairwise alignment. So this is for the guide tree formation. So this is just an equation to relate this distance mate tree distance scores to your similarity scores for the pairwise alignment. Now, distance scores, you would see that in phylogenetic trees. Usually seen how far away the divergence of a certain mutation is based on the theory of evolution. Pardon the noise. Now, anyway, in this the formation, or when we do multiple sequence alignment, just like when we do pairwise alignment, there are if you remember, it's inevitable that we sometimes have to introduce gaps and extensions. Now, there is a rule called once a gap, always a gap rule from the Feng-Doolittle approach. The rationale for this one is that for the two most closely related sequences that are initially aligned, they should be weighted most heavily in assigning gaps. The reason is because we do not want gaps. Why do we not want gaps? Now, it goes back to molecular biology. If you remember, the way we translate the mRNA to proteins, we have codons. Codons in groups of 3. Now, imagine if you have an insertion or a deletion mutation. Starting from the site where the mutation, the insertion or deletion mutation occurs, you would have a shift in the codons, basically a shift in the reading frame. And when you shifted, when there's a shift, even it's just a 1 position shift, actually 1 or 2 position shift is the most dangerous. The 3 position shift is actually the safest, or any multiples of 3, because it's a codon is a group, we group them into codons. But anyway, when you shift it, we are actually shifting the whole, we would, starting from the site of mutation, we would be shifting the whole reading frame. Therefore, we would end up with a whole different type of, or rather with a very different protein sequences succeeding from that shift. And that is why gaps, introduction of gaps, even in pairwise alignment, has the highest, usually the highest penalty, because we don't really want to have a gap. Now, the once a gap, always a gap, is that if we do need to have a gap, extending that gap is, we would put a much less penalties from the scoring than introducing another new gap. So basically, this algorithm, for the progressive algorithm, once we now have a gap in our alignment, it's much better to just increase that gap, add more gaps from that already existing gap in order to get a proper alignment. Because, for example, you introduce a gap in that position, you still do not get a good match. So just extend. So you would open up a larger gap. It's much better than introducing gaps, single gaps every which way. So inserting just single gaps where we think it's needed. So just insert a gap and then extend it basically that's the rule here. So the thing here is it goes by also on the idea that when you have such mutations or some breaks, usually these insertion deletions can be a result of breaks in the DNA or portions of the DNA itself that are being cut off. So basically, instead of having one single gaps interspersed in the sequences, just group them all together or into a huge chunk. So that's a better let's say more optimal alignment that we would have. So we want it to be in that way. The gaps are exist in a chunks rather than in a single interspersed single gaps across your alignment. Okay, now that's for the progressive approach. Let's move on to the iterative approaches. So iterative methods compute a suboptimal solution using progressive alignment. Basically, it's an improvement on your progressive alignment, but instead of doing one progressive alignment, you are actually modifying this alignment that you have until you have a convergent solution. So you modify it to alignment until there isn't really much changes in the final resulting alignment. So that's your iterative approaches. So you have an initial tree divided and profiles from each side are realigned. So you the same way you do pairwise alignment, you generate a tree, but instead of adding them one at a time, you do multiple alignments, trying to see if there's a convergent solution. So you change it up a bit. This is a way to combat the problem that in the progressive alignment, that once you add a suboptimal solution or a suboptimal step in the progressive alignment, that suboptimal step, which i.e. a mistake, can be amplified or can persist until the final result. So you ending up with a result that's not really that best result. So in order to combat that, you rearrange and rearrange and rearrange. You do it again and again, and so until the final solution doesn't change anymore, and then you have your result in your iterative step. So here is your iterative method by MAF. So you have an initial alignment, and then based on the tree, based on this alignment, you switch around the tree. You do a different group alignment, and then you realign it again, and then try to do it multiple times until there's really no change. Basically, your solution has converged. So there's not much change in the final output, and then you have your result. Now, for the consistency-based approach, this adapt a different approach, but it also is built up from progressive alignment. But in this approach, it uses information about multiple sequence alignment as it is being generated to guide pairwise alignment. So, basically, when doing your multiple sequence, you do progressive, the initial tree, you align them first, and then align them again in a pairwise manner. So you do pairwise alignment again. So consistency-based techniques score pairwise alignments in the context of information about multiple sequence. So you adjust the score of xi to yi based on the knowledge that zk aligns to both xi and yi. So this approach is distinctive because it incorporates evidence for multiple sequences to guide the creation of pairwise alignment. So here is your pairwise alignment from one another, and then you extend it. So S1, S2. So basically, this is your tea coffee alignment results. So in here, you would have this, for example, this formation of these Fs here, S1 to S3 Fs. So in this multiple sequence alignment, you do a suboptimal multiple sequence alignment first. And then if you look at these regions where there is always F, so for example, there's an F here, but there's a C here. So probably it's not supposed to be aligned in this manner. S2 is not supposed to align in this manner with S1. Because S2 aligns well with S3 in another way, and S3 aligns in this manner with S3. Therefore, it would be better for this alignment, S1 to S2, to be shifted, because there is this misalignment in S1 and S2, but there's a good alignment here in S1 versus S3 and S2 versus S3. And you have this S2 C from S2, which is problematic. Now, why not just shift it so that if we look at the S2, S3 alignment, use it to align this S1 to S2. So shift it so you would have this new alignment between S1 and S2 where the F would no longer be with C. So here is the new alignment. And then you do it basically you redo basically this pairwise alignment, and then you are actually if you have a suboptimal alignment, you are aligning it the suboptimal or the problematic residue, because here the the problematic sequence here is S2. Now we can do you realign the problematic sequence so that it would have a better result in your multiple alignment. So basically that's your consistency-based approach. So you want to have a consistent alignment across all of the sequences. So the last one is the structure-based method. So structure-based methods, you are looking at the tertiary structures. Just a recap, what are tertiary structure? Proteins basically seen in proteins. Proteins, the three-dimensional structure of the protein is a tertiary structure. And these are mostly conserved. They tend to be conserved more than the sequences itself. So even if you have one or more substitutions in the amino acids, the whole shape doesn't really change much. So an example here is the human beta globin and the myoglobin. So they share limited sequence identity. Basically, we call it a twilight zone because it's in the border where it's not really acceptable as a good alignment. And yet, when you look at the three-dimensional structure, they are very, very similar. It's actually in the same manner in my image of the conotoxins a while ago. That's a structure alignment. So basically, the structure-based method is using this alignment from the structure to generate multiple sequence alignment. Now, the problem here is the amount of structures we have in the database. So the size of the database for structure is much less. If it doesn't really even amount to about 10%. It's less than 10% of the number of sequences we have for the primary structure, which is the sequences. So structure-based method might be a good approach, but it's really not that much feasible. That is why it's not much well known, because rather than finding structures for our proteins that we want to study, there are sometimes the structures does not exist yet. We still do not know the structure yet, because knowing the structure of a protein is a much more difficult than just knowing its sequence. And that's why we have more information, or we have more proteins that we know the sequence but we do not know the structure. And in order to do the structure-based method, we have to know the structure first. And that is why it's not really that well known. So the limitation for this one is that you must have the structure of the proteins you want to study. So anyway, this is your structures. Now, I will stop here, and then in the next part of the video, we will look at sample results from the different algorithms we have discussed so far.